Hello everyone. So today we were discussing the question that has appeared in GATE 2018 Life Sciences paper. The question number is 16. So the question uh, that states a polymerase reaction is carried out for 10 cycles in a volume of 1 ml with 5 molecules of template DNA, assuming that the efficiency of the reaction is 100%. The number of molecules of DNA present in 100 microliter at the end of the reaction is correct to an integer number. So this question is from the technique called PCR, or we can say polymerase chain reaction. Okay, so polymerase means to polymerize some uh, segment of the DNA. Okay, and it will be a continuous chain reaction. So that's how the term is given, polymerase chain reaction, or in general we call this PCR. So first let's start talking about this PCR reaction, the basics of this PCR. So PCR is a very generalized and basic technique which we are using in our laboratories. So why we use PCR? So PCR is being used to amplify a certain segment of the DNA. Okay, if you have a sample DNA and from there you want to amplify one particular segment, that's when you can use this technique to amplify that particular segment. Okay, so suppose you have the entire sample. Okay, you have this DNA sample with you, entire DNA sample in a small amount, I would say. Okay, suppose 10 microliter of the sample you have, 10 microliter sample. Okay, first, it is a very less sample. Second, it is not a desired fragment of DNA for you. It is a whole lot of extra pieces of DNA which is not required for your work. Okay. So, suppose you want to only one particular segment of this DNA. Suppose this is your particular gene of interest. Okay. This is one particular, like I would say, genomic DNA from certain organism. It has the whole large DNA piece, and this is one particular your gene of interest. This fragment contains your gene of interest and you want to amplify this particular gene of interest. So that can be done using PCR. Okay, so at the end of the PCR reaction we'll have many fragments of this particular gene of interest. Okay, there will be many fragments to it. Okay, from the whole piece of DNA we'll have this particular fragment amplified. So that is why you use PCR reactions. Okay, so let's first talk what are the components that you require to carry out the PCR. Okay, so the components of PCR, the very first thing that you should have the sample DNA, or I would say the template DNA. Okay, this is the first thing. Okay, now if you'll have the template DNA, the second thing which you require is, so because in this no in this particular technique we use an enzyme which we call as polymerase enzyme. to carry out the polymerization reaction. So you need a polymerase enzyme. So the polymerase enzyme needs a 3' end to extend that particular DNA segment. Okay, so this polymerase enzyme needs a free 3' end. Okay, as we know, if we have a DNA sample, it has a particular direction from 5' to 3'. Okay, and this particular polymerase enzyme need free 3' end so that it can extend the DNA fragment. Okay, so to provide this free 3' end, we need a primer. Okay, so a primer is a short segment of the DNA. Okay, so if I'll say that, if I'll initiate, this is my particular gene of interest and this is my another extra pieces of DNA. So the first will be the breaking of this hydrogen bond. Okay, so the entire, let's draw this, the entire hydrogen bond is being broken. Okay, so the two DNA strands get separated and you want to amplify this particular segment, so you have to provide the primer. Okay, the primer pair which will bind to each of these strand. Okay, so this primer will bind to your template strand and here it will be provided the 3' free 3' end will be provided. Okay, so after the primer is being attached to this DNA template, your polymerase enzyme can sit here and now it can extend. Okay, in 5' to 3' direction, this fragment of DNA can be extended. And next, for extending this particular fragment of DNA, what you need the complementary base pair or the complementary nucleotides. So which can, those nucleotides in the form of DNTP. So you need deoxynucleotide triphosphate. So you need many different, all these mixture. So these DNTP mix you will be needed. So these DNTPs will be added here according to their complementary base pair. Okay, so this four basic things you will need, and to functioning, for the functioning of this polymerase enzyme, what else you need is a buffer which has particular pH, which has particular divalent cation concentrations. Okay, so that in this particular pH this polymerase enzyme performs its optimal activity. Okay, so these are the components that are required for the PCR to carry out. Okay, so now we have our whole components list. Okay, so as we have already mentioned, so suppose this is my double stranded DNA. Okay, this is my double stranded DNA. The first step is the denaturation. Okay, the first step of the PCR is denaturation. Okay, first we need to separate these two strands. Okay, and after the separation the primer will attach. Okay. The primer will attach. Okay, and then the polymerase will bind and extra those DNTP will attach and this particular fragment will be amplified. Okay, these fragments will be amplified. So now from one piece of DNA you have two pieces of DNA. Okay, and in the next cycle, so this is the first cycle, and then in the second cycle, now again these two stand. So this was a whole big chunk of DNA. From there we have given the gene specific primers. Okay, so that is why only one particular, this is a, okay, so according to the primers only particular region is being amplified. Okay, so again the above strand will be denatured, the above strand is denatured, above strand is denatured. Okay, now again the primers will bind. Again, so this primer binding is called annealing, annealing of the fragment. Okay, so again this will be synthesized. So now from 2 you have 4 pieces of DNA. Okay, so that is the whole thing that is being amplified. Okay, so the first thing you are seeing that you are having a denaturation step, you are having, and I made it together. So this is the second step, which is annealing, which is also you can call primer annealing, and this is the third step, which is extension. Okay, and after that again you will go back to denaturation. Okay, again denaturation, again the second step, and again the third step, extension. This is the first, second, and third. Okay, so these three processes keeps on happening in continuation, one by one. Okay, in one cycle. Okay, so what is needed here to carry out these reactions is the temperature. Okay, so the one cycle is complete, again you have to provide. So for the denaturation what we are doing, so for the denaturation what we are doing, we are providing a high temperature to the DNA. So upon getting the high temperature, both the DNA strands are being separated, the hydrogen bonds in between these are being broken, and the DNA strands are being separated. And then we are, okay, so we have a high temperature, that is suppose 90 to 98-degree temperature for the denaturation. Then we are supposed to give a certain temperature for the annealing. So annealing temperature is generally lower than, it is not up to the denaturation temperature, it is somewhere lower. Okay, so this is the annealing, or you can say primer annealing. Okay, annealing temperature, so annealing temperature will be Tm minus 5 degree, so Tm of the particular primer. So as I have said that primer are the short fragments of the DNA, okay, short oligos, so they are generally 15 to 25 uh, nucleotides long. So accordingly we calculate the melting temperature. Okay, so if we have A and T nucleotides present, it will be of 2 degree each. If we have G and C present in this oligo, it will account for 4 degree temperature. So accordingly we can calculate the melting temperature of the primer, and whatever the melting temperature we get, we'll deduct 5 we'll subtract 5 degree from there, and that will be our annealing temperature. Okay, so this will be our annealing temperature. Accordingly, we set the annealing temperature. Okay, now after the primer are getting annealed, we provide the optimum temperature for the polymerase activity, that is generally in range of 68 to 72 degree. So this is for extension. Okay, this is the extension temperature, that is optimum temperature for the polymerase activity. Okay, so that is what happening in the first cycle. We have the denaturation, then the primer annealing, and then extension. Again, for the next cycle we have to repeat this step. So this step we repeat several times. Okay, suppose 30 times we are repeating, so same steps it needs to be repeated, and after the repetition is done, we give one final extension and we put our reaction to the hold. Okay, so to carry out this fast changes in the temperature, we need the PCR machine, or also we can say thermocycler. Okay, so a PCR machine is nothing but a thermocycle which can perform the sudden change or the quick change in the temperature to carry out this PCR reactions. Okay, and this uh, so this is the basics. So to summarize, we have the PCR components that are needed in a thermocycler are the DNA samples or the template DNA. We'll be requiring the primers so that it can so that it will amplify the particular specific segment of the DNA. We should have the nucleotides so that during the extension step, the nucleotides will be added to make the complementary base pairing and to synthesize the new strand of the DNA. We need Taq polymerase to perform the function. So Taq polymerase which generally purified from the Thermus aquaticus organism. So and this is thermally stable. Okay, because as I've already said that this denaturation step we are performing at very high temperature, so this polymerase which we are using, it should bear the high temperature. So this Taq polymerase is thermally stable, so at the high temperature also there is no loss in the activity of the Taq polymerase. Okay, we should have a mixed buffer. All these things will be added into the PCR tube, and the PCR tube which is kept in the thermal cycler, and this thermal cycle do the quick changes of the temperature, and inside the tube we'll have this whole PCR cycle where we have the first step, the denaturation, which separates the strand. Seconds, the annealing step where the primers are binding to the template DNA, and then using the 3' end of the primer, this Taq polymerase enzyme is adding the complementary nucleotides to synthesize the new strand of the DNA, that is called the extension steps. So this is the one cycle. Okay, this is the one cycle. Similarly, this step will be repeated several times inside the thermal cycler to get the enough amount or the amplified amount of the DNA segment. Okay, so this is what we wanted. So this is the template DNA. This red portion is the specific gene, and we provide the primer for this specific gene only. So in the first cycle, from 1 we have 2 DNA fragments. From in the second cycle, from 2 we have 4 fragments, and the third cycle, so it goes on. So we have 16, then we have 32 fragments. So it increases exponentially. Okay, so it is an exponential amplification. So if we'll start from 1, so this is the formula to calculate the final number, final copies. N is number of cycles. Okay, this is the initial number with which you have started, initial number of fragment, and this is the final fragment, how much you will get. And this is the formula using which you can calculate, if you start from a particular number of DNA, at the end how many DNA you will be having in your reaction. Okay, so now if we look at the question, so our question states that the polymerase reaction is carried out for 10 cycles. So here n is given, n is 10 cycles in a volume of 1 ml. We have the volume, total volume is 1 ml, or we can also write 1000 microliter. So with 5 molecules of template DNA. Okay, so we have 5 molecules. Okay, so this is our initial molecules. 5 molecules of the template DNA. Assuming that the efficiency of the reaction is 100%, the number of molecules of DNA present in 100 microliter at the end of the reaction. Okay, so as we have discussed about that formula, so the formula that states final molecules is initial molecules into 2 to the power n. So this, we have initial 5 molecules, we have total 10 cycles. So we have total 10 cycles. So if we'll solve this, we'll get a number which is so we'll get a number which is multiplied by 5, we'll get, okay, so these are the final molecules. So this are the final molecules in 1 ml reaction. Okay, and it has been asked how many molecules will be present in 100 microliter reaction. So 1 ml, or we can also say 1000 microliter. And so if in 1000 microliter we have 5120, then in 100 microliter we'll have 512 molecules of DNA. Okay, so the answer will be 512. So this will be the answer of this question. So these are my NPTEL references. Thank you for listening.